morning, everyone. I'm Kevin Bennett. I'm a graduate student at the University of Maryland in Mike Braun's lab at the National Museum of Natural History. I'm going to talk about some pretty recent work that we've been working on about looking for the genetic basis of sexually selected traits in a hybrid zone. So sexual selection has produced some of the most amazing and beautiful and fascinating traits that we see in nature. Amazing colors and behaviors and structures. And scientists and non-scientists alike have been really interested in this process for a long time. As we know, Darwin wrote a book on the subject, right? And it's still to this day generating a lot of discussion and study over questions like the role of sexual traits as honest signals of fitness. But despite the fact that we've been interested in these for so long, we understand the genetic basis of almost none of these traits. And one reason for that might be that it's just hard to find situations where these kinds of, where the genes causing them can be teased out. But the system that I'm going to describe for you, I think is uniquely suited to do that. So the genus that I study is called Manicus. This is in the manakin family of birds, which becomes sort of a model system of sexual selection because of their varied and elaborate courtship displays. So in this genus, there are 4 members going clockwise from this bird. You've got Manicus, which is the white-bearded manakin, and then Arentiacus, Candii, and Vitellinus, which are the orange-collared, white-collared, and golden-collared manakins. And they can be thought of as sort of a superspecies. So they replace each other geographically, and everywhere they come into contact, they form a narrow, stable hybrid zone. They also share breeding biology, so they're like mating birds. That means that the males gather in one area and make display courts and display to attract the attention of females. So I'll show you what this looks like in white-collared manakins. They snap their wings together to make these buzzing and snapping sounds, and then jump back and forth between little sapling trees that make up the perimeter of their display court, while the green females will watch. So females will come to elect, evaluate different males, and then potentially mate. And work has shown that there's really uneven mating success across these legs. So one or a small number of males will get most of the mating. And that means that there's an opportunity for really strong sexual selection to happen here. So just to zoom in on the range map that I showed you earlier, here in western Panama near the Costa Rica border, if you were making your way through these numbered populations that our lab has studied, you'd be encountering golden-collared manakins, golden-collared manakins, golden-collared manakins. You wouldn't really notice that much of a difference as you went west, except maybe that the collar got a little bit paler yellow, maybe a little bit less olive color in the belly. And then you go across the Rio Changuinola and you find white-collared manakins. You would probably say to yourself, well, here's the taxonomic boundary. And these measurements of plumage color would tend to agree that there's this sort of steep transition in both belly and throat color right at this dashed line, which represents the river. However, it turns out that in a suite of genetic markers, both nuclear and mitochondrial, it turns out that the genetic transition between these taxa occurs about 50 kilometers to the east between numbered populations 8 and 9. So what we actually have going on here is golden-collared manakins up to this narrow hybrid zone. And then in between the hybrid zone and the river, you have birds that genetically resemble white-collared manakins, but show the golden-collared phenotype. And then on the other side of the river, you have typical white-collared manakins. And what's happened here is the golden-collared phenotype has introgressed from golden-collared manakins into white-collared manakins through positive sexual selection. So we know that golden-collared males are more aggressive than white-collared males. They're more likely to attack a taxidermy mannequin put on their display court. And then also, in the headwaters of the river here, where you can find both golden and white-collared males in the same lek, females will preferentially mate with the golden-collared males. And so the trait is introgressed into white-collared populations up to the point of the river. So an important question for our lab, and something that I was interested in working on when I started my PhD, is what gene or genes are responsible for this color change? And we don't know as much about the genetic basis of yellow color as we do about melanism, for example. We don't have these great candidate genes like MC1R to go look for. But we had some idea of the kinds of genes that we were looking for. So maybe genes related to carotenoid deposition. If you know about carotenoids, they're these yellow pigments that are often the cause of yellow and red colors in animals and plants. Other genes we might expect could be those related to regulation of feather development or regulation of gene expression. So here's what we did. we sequenced the genomes of 26 individual birds across 4 populations. This is data that was collected by H.C. Lim, who's a former postdoc in our lab, now a professor at George Mason University. Population 2 and 12 are our allopatric, white-collared, and golden-collared populations. And then 3 and 4 are both genetically white-collared, but they're on either side of the river. So population 4 has golden collars, population 3 has white collars. So we sequenced these genomes, lined them up to our reference genome that we have for the golden-collared species, and then identified variants, SNPs and indels. And we got over 20 million high-quality SNPs across these 4 populations. So then I could go through and calculate FST, this classic measure of genetic differentiation, between populations 3 and 4. So where in the genome are they different from one another? But then you can also use the information from these parental populations to try to find introgressed variants. So what sites in the genome show this sort of expected pattern of having introgressed from golden-collared mannequins up to population 4, but then not yet reached populations 3 or 2. And here's what the data look like. The top panel is FST. The bottom panel is the frequency of these potentially introgressed variants. Each point is a 10 kb window, and then alternating colors are just different scaffolds from our reference assembly. And a few things you might notice right off the bat. In FST, there's this really low level of sort of background genetic differentiation between the populations, which is what we would expect since they're really close to one another in space. You might also notice that there are a few scaffolds that have sort of somewhat elevated FST across the scaffold. And it turns out that most of these fall on chromosome Z. So this is a sex chromosome in birds, has a smaller effective population size than the autosomes. And so it's predicted to have, to be more susceptible to the effects of genetic drift. So we haven't confirmed that that's what's going on with these, but that's what we suspect. And then finally, you might also notice that there are these 2 pretty nice clear peaks in both FST and the frequency of these introgressed variants. One of them corresponds to a site on chromosome 2, the other one corresponds to a site on chromosome 24. So I'll show you that chromosome 24 peak. This is what it looks like when you blow up the scaffold. Really clear peak in both FST and these introgressed variants. If you go and look at where they're clustered, which is basically the center of that FST peak, you find a gene called beta-carotene oxygenase 2. So beta-carotene is a carotenoid. I mentioned earlier that these yellow pigments. The activity of this enzyme, BCO2, in the cell is to take carotenoids and chop them up into colorless products. So I knew I had a really good candidate right here, but it turns out that there are a number of instances in nature where this gene has been related to a white-yellow polymorphism. So in wild red jungle fowl have white or gray skin. Domestic chickens have yellow skin. The reason for that difference is that BCO2 is not expressed in the skin of domestic chickens. Another example you might be familiar with, this golden-winged and blue-winged warblers in North America are really similar to one another across the genome. Where they're different is in 6 peaks of differentiation. One of those is over BCO2. So it's not just in birds where we've seen this before. The food industry has identified a number of other examples. So in sheep fat, cow milk fat, and rabbit fat, a loss-of-function mutation in BCO2 causes carotenoids to build up in the fat, and so you get this yellow or yellower fat phenotype. So I think all of this evidence together is really strongly suggestive that BCO2 is an important component in our story. And even though we still don't understand as much about the genetics of yellow coloration as we do about melanistic polymorphisms, I think we should start thinking about BCO2 as maybe the MC1R of yellow coloring animals. The last thing that I want to say about this enzyme is that it's expressed in the mitochondria, and at least in mice and humans, we know that it's involved in regulating oxidative stress. So it may be that by the time we learn a little bit more about it, what it's doing and how it's involved in these mannequins, we could be able to say something about the role of yellow as an honest signal of fitness. Okay, so I showed you there were 2 peaks. The other one is over these 2 really tightly linked genes. I'll show you both of them. The first one is called R-spondin2, or RSPO2, and this is an activator of Wnt signaling. So Wnt has a bunch of different roles throughout the cell, but it's been shown again and again to have a special role to play in regulation of feather development and also color patterning in a bunch of different animals. So heliconius butterfly wing patterns are partly controlled by one of the Wnt genes, Wnt A. If you saw Anna Vickery's talk earlier in the meeting, you've seen this plot before, but the difference between two color forms of domestic pigeon is this other Wnt activator called NDP. And then finally, this warbler story that I already showed you, where these two warblers are only different at 6 points in their genome. One of them is BCO2, another one is one of the Wnt genes. So again, I think this is very strongly suggestive that this gene is important in this color polymorphism of mannequins. So there are two genes under that second peak. The second one is called eukaryotic translation initiation factor 3 subunit E, or EIF3E. And this one, we're still sort of exploring how we think this is involved, but it's a little bit tantalizing because the more we study regulation of gene expression, the more we start to learn that regulation of gene expression is controlled not just at the transcription level, but also at the translation level. And EIF3 and its various subunits are some of the most common, or maybe the most common, targets of tissue-specific and pathway-specific regulation of gene expression through controlling translation. So if you have any insights into this, I would love to get your feedback about it. Okay, so just to sum up, the different color forms of white-collared mannequins are very similar to one another, but where they're different, the most different, is in these three genes, one related to carotenoid metabolism, one related to feather development, color patterning, and then another one related to regulation of gene expression. And there's a lot left to do. This is all sort of very new stuff that we've just been working on in the last few months, but in the future, I'd really like to be able to find the causative variant at the sequence level. I didn't mention this before, but there are no amino acid changes in the pattern that we would expect between white and yellow birds in these genes. And so I'm probably looking for something regulatory. Really like to look at expression differences in these genes between white and yellow birds. And then just more generally understanding the molecular mechanism. So how are these pieces that I described, how do they fit together in the cell to turn a white bird into a yellow bird? I think we've answered an important part of that question, but there's still a lot left to do to get the full answer. So to conclude, I'd like to thank many supportive people at the University of Maryland and the Natural History Museum, as well as the Mannequin Research Coordination Network and our funding sources. And thank you very much for your attention. Time for questions? Yeah.
Well, what do the hybrids look like? Are they intermediate?
The hybrids are, they look like the golden collared species. The closer you get to the river, you get a little bit, I can show you the clans again, you get a little bit of color change, like sort of gradual color change where the collar is just a little bit paler, but they pretty much look like the golden collared birds. So you can see here, the clans aren't quite as steep as the genetic clans are, but they're pretty much, they look like the golden collared birds. Maybe just less olive and, yeah. Question.
Do you have any idea for genetic basis of female preference for these colors?
That's a great question. It's not something that I have any insight into yet, but it's obviously a really important component. So that's definitely something that we're going to be exploring. Yeah. All right, thanks everyone.